I am now pleased to present this year's ASM lecturer, Dr. Frances Arnold. Dr. Arnold is the Dick and Barbara Dickinson Professor of Chemical Engineering, Bioengineering, and Biochemistry at the California Institute of Technology. Her research focuses on direct evolution of enzymes and biosynthetic pathways with applications to alternative energy, chemicals, and medicine. Her work pioneered directed evolution methods that are used in laboratories throughout the world to make products that range from medicines to foods to textiles, chemicals, and fuels. Dr. Arnold received her bachelor's degree in mechanical and aerospace engineering from Princeton University in 1979. After a year working as an engineer in solar energy research in Brazil and Colorado, she enrolled as a graduate student at the University of California at Berkeley, where she earned her doctorate in chemical engineering in 1985. After postdoctoral work in biophysical chemistry at UC Berkeley, she joined the Caltech faculty in chemical engineering in 1987. Dr. Arnold is the recipient of numerous honors and awards, including the 2011 National Medal of Technology and Innovation and the 2011 Charles Stark Draper Prize of the U.S. National Academy of Engineering. Dr. Arnold has also been elected membership to all 3 National Academies of Science, Medicine, and Engineering, and was elected to the American Academy of Arts and Sciences in 2011. Among many other activities, Dr. Arnold serves as a judge for the Queen Elizabeth Prize in Engineering, and we almost didn't get her for this talk because of that. The President's International Advisory Council of the King Abdullah University of Science and Technology, and on the advisory committee to the David and Lucille Packard Foundation Fellowships in Science and Engineering. She has co-authored more than 250 publications and is the co-inventor on 38 patents. Dr. Arnold is also active in technology transfer to the private sector. She has served on the science advisory boards of numerous companies, and she co-founded GevCo Inc. in 2005 to make fuels and chemicals from renewable resources. Please join me in welcoming Dr. Arnold.
Thank you so much. I think you all are going to have a wonderful 4 days here in Colorado. So much exciting science, and this session so far has opened it beautifully. I found myself getting chills listening to some of the material that was presented earlier. I'm really thrilled to be here, and I want to thank you for the opportunity to address this audience. And I also want to thank Nathan Wolfe for that marvelous introduction, and especially for this theme of exploration. I want to follow up on that idea of where will we explore in the future, and I hope that I will leave you with the idea that there's plenty of room for explorations. Now, as you've heard, I am a card-carrying engineer. But I stopped working on man-made systems, even though I felt that aerospace was a very interesting area of being able to build fascinating and useful new things. And I moved into the chemical world and the biological world many years ago. I think we have a lot to learn as engineers from biology, because you only have to look at some of the things that Nathan showed and some of the things that you work with every day to realize that when it comes to being able to extract energy and materials from the environment and convert those into amazing, self-reproducing, self-repairing machines, there is no one better than nature at engineering such things. And when you think about the incredible diversity of resources that the microbiological world can use, that the entire natural world can use, you realize that there's an immense area of exploration for us to learn and become inspired by when we build new things. So I think this meeting is a great place for engineers to come because we can get this inspiration that we can use. Now, I'm not going to give you a lecture on the revolution in molecular biology that's happened in my lifetime. I graduated with my PhD right pretty much at the beginning of the time that we could manipulate the DNA that encodes all these functions. I'll just remind you of what you already know, that this technology has moved so incredibly quickly that in this 30 years, basically only 60 years since the discovery of the structure of DNA, about a little more than 30 years since the first genetic engineering experiments were performed. But all this functionality, right? We hear about sequencing, we hear about understanding the DNA sequence that encodes all this amazing functionality. But now we're at this point in the technological development that we can also rewrite this DNA, recode it. Now, the synthesis of DNA, you can also see has moved very, very quickly, such that you can write down a sequence of DNA, mail it off by email to your favorite supplier in Northern California or in Taiwan. In a few days, you will get back in the mail the actual genetic material. Absolutely amazing. And this has allowed researchers like myself, but many researchers to go and recode organisms to do an amazing variety of things. Perhaps you'll remember the headlines from a couple of years ago where the Venter Institute synthesized the entire genome of a bacterium, perhaps not a terribly interesting one for many of you, but an entire genome nonetheless, from non-living material. That synthetic DNA was incorporated back into a related organism whose genome had been degraded, and that organism rebooted. It read that DNA as if it were its own, and it continued to reproduce and do whatever that organism does. A truly remarkable demonstration of our capabilities now to reprogram the microbial world. But here is the fundamental problem, and this is the problem that has fascinated me. Craig Venter did not compose that genome. He did not write that music. He copied it from non-living material, and he put it back into an organism. But the composition itself, the genome, to me is like a heavenly piece of music, intricate, beautiful. We don't know how to compose like this. We are just learning to hold the pencil when it comes to writing out the DNA that would solve human problems. This is what an engineer does. We want to solve human problems. We want to use the biological world for inspiration to solve human problems. But all we can do now is pull chapters off of Google and put them together and hope that makes good music. A little bit of bits and pieces of passages from various pieces of natural music and hope that that new combination sounds good, but it doesn't. Sometimes they function, but they don't really do anything all that useful. And so that's the challenge that people like me face. And if you were going to go out and use advances in molecular biology to rewrite the code of life, you too will face this challenge. I work mainly with proteins, not even the whole organism, just one tiny element. And I love enzymes because they're the ones that convert one stuff into other stuff, and we do need a lot of stuff. And microbes, biology is a pretty good way of making stuff. non-toxic, sustainable fashion. And so people like me would love to be able to reprogram even individual proteins so that they can, for example, catalyze reactions that would be useful to us. But how do we do that if we don't even understand how the sequence of a single gene encodes a folded protein? So we know how the DNA encodes a polypeptide chain. That problem was even solved before I was born, and that was a long time ago. But when I was a graduate student, and some of you in the audience, you should be thinking about this. When I was a graduate student, they told me the protein folding problem would be solved in 5 years. They lied. The protein folding problem has not been solved. 25 years later, 30 years later, we still don't understand how the sequence of amino acids encodes a folded protein. And you know what? Who cares what the folded structure is? What really matters to me is what does it do? So there are two great big questions that remain. And if I, as a protein engineer, I want to go in and write DNA that encodes new protein functions, I've got a fundamental problem there, which scared the bejesus out of me as an assistant professor until I realized that I really didn't have to know in my big brain, actually my very tiny brain, the answers to those questions before I could engineer the biological world. Because there's an algorithm that does it very, very well: evolution. All you have to do is look at the sequences and structures of proteins that exist today to build family trees that take you back to some place in the distant past where that protein had come about and started to diverge into families that are all beautifully tuned for their particular biological roles. Every molecule in these families does a job and does it quite well. And it all came about not through answering those red question marks, but through a simple algorithm. An algorithm, you can crank it out, of mutation and natural selection. We've been doing that for a long time, actually. We've been using that algorithm to engineer the biological world. You didn't need molecular biology to create new forms of life. Well, not really new forms of life, but to modify existing forms of life in highly non-natural ways. None of these things would be fit if released into the natural world, but they're useful to us. And so for thousands of years, we've been modifying everything from corn to carrier pigeons, lab rats, racehorses, in order to solve human problems. Now, of course, with molecular biology, we can speed that up a bit, and we can get beyond the limitations that faced the farmers in the past. So the animal breeder was limited by the fact that pretty much monkeys go with monkeys and worms with worms, and never the two shall meet. But now, imagine we've got all this capability. You can synthesize anything you want. You can breed 3 creatures, 33. You can dial in as many mutations as you want randomly, or you can direct them to a particular place. In the test tube, you don't have any of the limitations that faced us in the past for breeding. This is, of course, great news for the explorers, right? Because there's a big place to go and explore. And of course, it's also a big challenge, because you can imagine that when you willy-nilly put DNA codes together, or take genes and cross them in different ways, most of the things that you will make will not be at all interesting. They won't be functional, and they certainly won't solve a human problem. But here's my exploration slide. Nathan told you that there's a whole world of organisms out there. In fact, organisms that we don't even know exist, but will be open for others to discover. I will tell you, there is a whole space, essentially infinite, of DNA sequences that nature has never explored, probably never will, but they're open to you. And in that space, I tell you that there lies the answer to the energy crisis, the cure for cancer, maybe even a cure for old age, who knows. There are lots of things in that space of physical possibilities. And I also tweak the biologist. Most of you study the natural world. You're in that little green space there. Those sequences that are relevant to biology today in 2013 are the products of natural selection, and they're tiny, tiny, tiny fraction of the possible things that could be made. So do not worry. There's lots and lots of sand in this sandbox for people to play in. Now, before I get into how do you evolve new things in the laboratory, which is the topic of my presentation today, let me tell you that it's actually pretty hard to do, and I hinted to that a little bit. Consider just a single protein, 400, my favorite protein, heme domain of a cytochrome P450, 450 amino acids long, 20 amino acids in nature's alphabet, give or take a few. That gives you a space of possibilities 20 to the 450th power, which you cannot even begin to comprehend the size of. Many, many, many times larger than the number of particles in the universe. This is an enormous space just for a single protein. And I will also categorically tell you, even though I haven't explored but the tiniest fraction, that it is mostly empty. Most of those sequences don't encode a protein that does anything, and they certainly don't encode a protein that will solve your problem. So the real challenge for the evolutionist is, and for anybody exploring that big space of physically possible proteins, is how do you get there? How do you get something going out into that space that will be useful, that will be beautiful, that will write a code of life that will be pleasing and solve a problem? I will condense a whole set of lectures in evolution down to one illustration. It works because we're here. John Maynard Smith, long before the sequences of proteins were known, and you could actually see how mutations were readily accumulated in proteins, argued that proteins have to form a vast connected network where moving from one sequence to another by single mutations can't be always deleterious, otherwise we would not evolve. And we are the products of evolution. Well, now with all these wonderful sequences that we can see, and with experiments that we can see both in the natural world in antibiotic resistance or virus evolution, and in the laboratory, such as I will describe to you, we can see that proteins can accumulate mutations. Sometimes they're deleterious, sometimes they're neutral, and sometimes they adapt. They allow you to climb a fitness peak and become better at something. So at least in some of the dimensions that you can travel in, that being a direction, a change in a sequence going in a particular way, we can walk one step at a time and accumulate mutations and, in fact, make proteins that are better by my definition. Remember, I'm the breeder of proteins, like all those strange-looking dogs that may not be fit in the natural world, but they're better for me. So I call this process directed evolution because I decide the fitness criteria. But I have to start with something, and that's where the natural world, that little green space, is very important. I can find those rare functional sequences. Out of all the possible proteins, I can find the ones that encode a folded functional protein simply by scraping it off the bottom of my shoe. It's given to me on a silver platter as the result of billions of years of natural selection. In the laboratory, and your kids, if you aren't doing these experiments yourself, your kids in high school are doing these things, you can just PCR up a gene sequence And if you do it under conditions where the polymerase is a little bit sloppy, you can make lots and lots of mutants. It's very simple to make randomly mutated proteins. The cells then do all the work. They translate that it's very easy to make randomly mutated genes, and then the cells do the hard work to translate that into randomly mutated proteins. That's where microbes are my best friend. And then I have to do the hard work to decide who is better now by my set of criteria. So we will ask lots of questions, and we will do 1920s German analytical chemistry and high throughput, measuring the products of certain reactions catalyzed by enzymes, for example. And we will find out what everybody has found out in the past, that the vast majority of mutations don't improve your protein. And in a simple evolutionary algorithm, random uphill walk, those get thrown away. But if you've asked the right kind of question, and this is the real key to evolving something in the laboratory, just as it is in animal breeding. If you've asked the right kind of question where the improvements can be obtained through small changes, typically a single amino acid substitution, and you can measure those small changes, then you can accumulate beneficial mutations over multiple generations, acquiring the properties little by little until you've met your goal, until your grad student gives up and graduates, or whatever it marks the end of your experiment. This is actually an algorithm that works incredibly well. Hundreds of laboratories use these methods now, and it seems so obvious now, but this was not the paradigm for protein engineering for a very long time. People really did believe that they could answer those red question marks and predict the effects of mutations. But you don't have to. You can use this evolutionary algorithm. Here's a beautiful colored example of one of the first adopters of directed evolution methodology, Roger Chen, who took those lovely green and red fluorescent proteins from the natural world, from jellyfish, and mutated them through directed evolution and made this whole palette of beautiful colored proteins just by diversifying it. And he decided who lived and who died and who went on to parent the next generation. And of course, these are now ubiquitous. Everybody uses them, and we all benefit from the products of this, of Roger Chen's work. Now, as you heard, I'm a chemical engineer. I've been focused on using microbiology to make fuels and chemicals that we currently get by pumping it out of the ground. Someday, I don't know when, someday we will have to stop doing that, either because our planet becomes way too hot and we can't tolerate pumping CO2 into the atmosphere, or it's simply, it all comes down to it becoming far too expensive. We want to find alternatives, and this is where my inspiration from the natural world comes in, because that's what microbes do. They convert energy and matter into more microbes. My job as an engineer is to convince them to stop making more microbes and have them make the products that we need. My favorite protein, as I mentioned, is the cytochrome P450, and I'm going to use this as an example of these methods, from Bacillus megaterium. That's a soil microbe. You literally can scrape this beautiful molecule from the bottom of your shoe. Now, chemists in the audience, there might be a few of you who love chemistry. Otherwise, don't leave yet. I won't be hitting you with too much chemistry. But if you're a chemist, this molecule, this catalyst, you will find incredibly impressive because it can insert oxygen into a CH bond at room temperature and pressure. No chemist has figured out a catalyst that can do that. Think methane to methanol. Nature does that, but no chemist has figured out how to do that unless you go to very, very high temperatures and pressures. It becomes extremely complicated and energy inefficient. But this is what nature likes to do. I work with this particular microbial version Because unlike the P450s that you have in your liver, for example, which are membrane associated and nobody would touch with a 10-foot pole in biotechnology, just evil to work with, this one's soluble. It's expressed in E. coli, and it's highly active. It's about 1,000 times more active than your own enzymes. Furthermore, it has everything it needs for its reductase domain, all in a single polypeptide chain. So even engineers like me, we can work with this. From the chemistry point of view, it activates oxygen in the heme. So it has this iron. It's trapped in iron in the heme. And chemists know that iron is a wonderful and versatile catalyst. Well, nature figured that out a long time before we did. And this particular enzyme from Bacillus megaterium likes to stick that oxygen that it activates at the iron into a subterminal position of a fatty acid. Well, nobody cares, right? It's not useful to hydroxylate fatty acids. So people have ignored this enzyme for a very long time, other than using it as a model system for understanding the mechanism. So this is a useless reaction from a chemistry point of view. But if you could convince it through directed evolution to do something else, for example, take natural gas and turn it into liquids, that could be useful. So starting now a long time ago, that's exactly what we did as an illustration of how rapidly a protein can evolve. This is what was so surprising about this. Over generations, you could take a protein that was very good on a long-chain fatty acid, had a little bit of activity on octane. You could screen it for mutations that gave you higher activity on octane, making octanols. And guess what happened? As you improved activity on octane, it started using propane. Nobody thought a P450 could use propane, because whenever you go out to the microbial world and you look at propane monooxygenases, they were diiron enzymes. But then not too many microbiologists work with propane, and oxygen tend to explode and things like that. So there really wasn't a very good sampling of the biological world for these activities. But you can see how readily, in just a few generations, you could create that activity in the laboratory, starting with something that works on fatty acids. In fact, this enzyme is so good. Well, it has 23 mutations by the time we got finished with it. That's probably not the minimal set, because we did all sorts of strange things on our little evolutionary path. But it worked very well. And in fact, this enzyme that was evolved in the laboratory is as efficient on propane as its ancestral wild type is on its favored substrate, a fatty acid. And during the process of this, even though we bred it for activity on propane, it forgot that it was a fatty acid hydroxylase. So the final enzyme is almost as efficient, but has now lost its activity. And the starting enzyme had no measurable activity on propane. So I use this example just to show how a simple, stupid optimization strategy, a random uphill walk, can be used essentially in an algorithmic fashion to create something that's really quite novel. Now, I'm interested these days in hard problems. What's a hard problem? Well, all you have to do is go to the creationist literature and pick out any number of ones that you want. A hard problem is evolving something that's surprising, right? Something that doesn't have some little measurable activity, something that does, say, new chemistry. That would be the equivalent of a new species if you're an enzymologist. And we just were very lucky recently to find a nice example of that. We know that nature created new functions, right? It has to be a product of evolution. But it's very hard to catch nature in the act of creating a new species, or in the act of creating a whole new function in a protein. You have to be there at the right time to catch that. But here we have to speed it up a bit, so we have to contaminate our blind experiments with a little bit of knowledge. I try not to do that very often because the contamination usually goes the wrong direction. But in this case, the little bit of knowledge that was useful was the recognition, a chemical intuition, that an enzyme that can activate oxygen at the iron might also be able to form what we call carbenes, because we knew that heme chemistry, people had done biomimetic chemistry using hemes. Synthetic organic chemists had done things like this. So we asked, could this cytochrome P450, under highly non-natural conditions, actually do a whole new reaction that nature has never explored? Well, the bottom line is yes, or I wouldn't be telling you this. And in fact, you don't even have to look at the chemistry. All you have to know is that if you take a highly activated diazo species, which you don't find in the natural world, but we could always add it, and you add it to a double bond like olefin, like styrene, this enzyme can create 4 possible cyclopropanes. That adds a carbon across the double bond, whereas the natural enzyme added oxygen. And you can make 4 possible things. But because you've got this wonderful protein there, it can select what product it makes. And so, for example, a single mutation in this allows this enzyme to make 1 of the 4 possible cyclopropane products. This is the sort of thing that makes chemists just go berserk with happiness. Chemists love to be able to combine molecules, make 3 stereogenic centers, and make 1 product. That's worth a lot. And then you can use evolution to tune this, because you've got this protein that's doing the fine-tuning. The iron is doing the work, but the protein's doing the fine-tuning, so you can now make, for example, another 1 of the 4 products. We have lots of fun with this. But let's get back to the microbiology. These proteins work in a chemist's test tube. Could these proteins do this whole new chemistry? Cyclopropanes, by the way, are found in the natural world, but they're made through totally different enzymatic mechanisms. If you could genetically encode these new proteins and put them into microbes, you could genetically encode whole new metabolic pathways that involve these functionalities. I like this view that we can take all the accumulated knowledge from synthetic organic chemistry and import it into a microbial system if we were so smart that we could make these new enzymes. Well, it's not an easy thing to do because this cytochrome P450 has to be reduced. We used, if you saw my little note on the previous slide, you saw that little thing called dithionite. That is not a natural reductant. NADPH is the natural reductant. But we found that NADPH couldn't reduce our enzyme. And that's because nature had designed this system so beautifully that it's gated, that just having NADPH there would not turn the enzyme on. Otherwise, it would just be chewing up that expensive NADPH all the time. It has to have its substrate and your reductant before it'll turn on. Beautiful example of regulation. Well, that really messed us up until we realized that, well, gee, this P450, which has another atom on the other side of the iron, it's a sulfur there, absolutely required for oxygen chemistry. No longer required if you're going to try to insert carbon atoms. So we simply changed that for a serine, and lo and behold, the protein folded fine. It no longer inserted oxygen, but it started to really like to insert the carbons. We call this whole new enzyme family the cytochrome P411 because P450 means you have a peak at 450 nanometers when you have CO bound. Well, once CO binds to this, it now has a 411. So I am the mother of a whole new enzyme family that, in fact, works beautifully inside of E. coli. This is not a given, but we were very lucky. You can stick this enzyme inside of E. coli, and you can just lyophilize the cells, and the protein's very happy. But it's even happier if the cells are intact and not lyophilized. And in fact, you can get 67,000 turnovers. That is, it'll crank out its product 67,000 times on average, which is higher than the number of turnovers ever reported by any organic chemist for this important synthetic reaction. We were absolutely thrilled at this. All right, so I've used this piece of my talk to illustrate how even an engineer can build a new biological, genetically encoded object that can solve a human problem by an incredibly simple algorithm. This has demonstrated how readily proteins can adapt to new problems and via a simple uphill walk. Very tiny changes in the sequence can give rise to very different functions. I've shown you 23 mutations in one example and just a couple of mutations in another. And for those would-be designers who think we can learn enough to solve those red question marks, I will just point out that most of the mutations that give rise to changes in function happen, many happen in the active site, but many happen far away. And their effects are propagated to the functional part of these molecules through mechanisms that we are a long ways from understanding. It all comes down to details. In biology, the details matter. They really matter a lot. It's very hard to do physics like biology. Details, it's all in the details. And we don't understand those details. So now I want to switch gears. I want to talk about sex. I think it's that time of night where we need to have a frank discussion about this random mutagenesis thing. We all know that, yes, we accumulate mutations through mutagenesis and selection, but there's other search strategies that nature uses to innovate. And one of those search strategies, of course, is recombination. Just think about it. Maybe 500 generations of dog breeding can give rise to an incredible diversity in the behaviors and in the phenotypes of dogs. What's happening at the molecular level? And can we use this search strategy to do things? So I'm going to tell you a little bit about sex with molecules. We have a lot of fun with this. And we do a lot of explorations. Our starting material usually comes from databases. You can go out and identify the sequences of proteins in a family. And I'm going to only talk about homologous recombination. Recombination from proteins that may be evolutionary, very divergent from very different species, but they encode similar three-dimensional structures. So we go out and we identify genes and ask the question, what happens when you recombine genes from disparate species to create, if it's functional, what we call chimeric proteins? So this is that wolford, the wolf bird. Doesn't look like a wolf bird, but it might have genetic material from a wolf and a bird. What's this worth? This is another set of explorations, and I will summarize some of the things we've learned. We've been doing this for about 10 years now. We've learned a lot of interesting things, and we've quantified much of this, but I won't bore you with all the numbers. I'll just point out that, first of all, sex works because you can make a lot of mutations at once, but it's very conservative. If you make a lot of mutations at once by random mutagenesis, you will see an exponential decrease in the probability that your resulting protein even folds. Mutations are, in fact, deleterious, and random mutations are very deleterious. But homologous mutations, mutations that are acquired by recombining genetic material that comes from proteins of a similar three-dimensional structure, are much less deleterious. That's because they've evolved in the context of that three-dimensional structure, and all the really bad ones got thrown away by natural selection. So you're recombining an already filtered set of information. So if you take two parents and you recombine them, the most distant things with the most mutations will have the lowest probability of folding in function, and as you go towards the parents, that probability goes to 1. And if you can think in hyper dimensions, you can do this for 33 parents, and you'll still get the same answer. Recombination is conservative. But it's still deleterious, because if you recombine from very, very different things, so if you're 80% identical, maybe 1 in 10 will fold and function. But if you're only 40% identity, when you recombine two parents, it's very hard to find chimeric progeny, the children, so to speak, that even fold. That's why when you have sex with your mate, you can create functional children most of the time. But if you try to have sex with a worm, your children won't be viable, right? We see that in the laboratory and with proteins. It's the same damn story. And we want to look at the ones, the rare ones that are viable. So that's not a zero there. That fraction functional does not go to zero. There's still lots of proteins. It's just that the probability of finding them is low. Okay, so here's another example where I contaminate the experiment with knowledge. I'm going to recombine, but I have my sticky fingers on the wheel for this experiment. And I'm going to choose 3 parents just because I can. It could be 33, but 3 is just not 2. So I'm going to take 3 parents, and I'm going to cut them and choose the crossover locations. And I'm going to choose the crossover locations in a way that I think will minimize the disruption in the children. Still acquiring lots of mutations, but the 3-dimensional fold of the protein, that is, whether it has 2 eyes and 2 legs, will be similar to the parents. All right, and that requires that we ask the question, what is the swappable element of a protein? Nobody ever asked this before, because nobody could ever do it until you could synthesize anything you want in the test tube. But now we can do it, now we can explore, and we have to answer this question. If you think of a protein as a Lego block and you're swapping in and out all the red, blue, and green pieces, what do the blocks look like? So here's my answer to that. It's a very simple thing, and I'm sure you can come up with a better one. But we look at the 3-dimensional structure of the protein as a series of connections between amino acids that have to interact with each other. And those interactions help contribute to the stability of the folded 3-dimensional structure. But once you choose parents, well, first of all, let me explain that when you cut the protein into pieces, you will break a lot of those interactions because you will swap in mutations from different parents. But once you choose the parents, you can, there will be a bunch of interactions that are shared by both parents. Let's say there's 2. And if they're 80% identical, then about 80% of those interactions cannot be broken, no matter how you cut and slice the genetic material. So the more similar they are, the more things cannot be broken. The less similar they are, the more things can be broken. So we can just look at the sequence identity of parents, decide how many interactions can be broken, and then what we do is we design the crossovers to follow the rule, thou shalt break the fewest interactions. And you can actually program this, and you can use dynamic programming, you can find a global optimal solution for how you would cut that. And that's what we do. And what that does, if you look at all the possible amino acids in a 2-dimensional diagram, and you look at the dots, those are connections, that sequesters these dots into segments that should be the swappable elements of a gene. So here's the experiment. We had a lot of fun with this one. We took our cytochrome P450s, my favorite protein, and we took 3 bacterial versions. They were about 60% identical, still had about 200 amino acids different. That's a lot of mutations to put into a protein. Took those 3 genes, cut them into the pieces designed to minimize disruption, and made 6,561 children. We looked through those to find the ones that folded. We found a bunch that folded. In fact, about half of the children folded and functioned as P450s, and about half didn't. Great about this experiment is that you find the non-functional ones as well. Nature threw those away, and you can't even find them on the bottom of your shoe. You only find the good ones. But theorists love to have the bad ones, too, so we publish those alongside the good ones. And that way you can test your folding algorithms. The other great thing is that unlike sequences in the vast databases that you get from the Sargasso Sea or the rainforest and everything that are in your computer, these are in my refrigerator. All these recombined P450s. Half of them folded. That was 3,000 new P450s. When we did these experiments, the entire databases for cytochrome P450 was 3,000. We doubled the database of functional P450 sequences. And they're all quite different from their parents. So what do? Are they at all interesting? So the other part of the story, recombination is conservative, but it's also highly innovative. Sex is good. Sex is good for evolution. And you can see it in the laboratory with proteins. And I don't have time to go through lots and lots of examples, but let me just give you one that is actually useful. It used to be that drug companies would grind up rat livers. In fact, people still grind up rat livers in order to test how drug candidates get metabolized. Because that's what the P450s in your liver do. They metabolize all the things that you ingest, particularly your drugs. And sometimes they turn that drug into something that's even more biologically active or that might even kill you. So they need to know, when you have a drug candidate, what kind of metabolites can be performed. This is also very important for new herbicides and pesticides and things like that. Well, in this library of recombined P450s, we could find enzymes that mimicked human metabolism. So the bacterial enzymes had now acquired the same chemical reactivities as essentially almost all the human enzymes. And you can go and buy these enzymes now from Codexis. A lot of drug companies use them instead of grinding up rat livers. They now use these bacterial enzymes that have been evolved in the laboratory. Recombination also explores lots of properties. And when you collect lots of data, so we measured the stabilities, for example, of several hundred of the progeny sequences. And we found, as you might not be surprised by, that there's quite a distribution in those capabilities. The vast majority lie within the properties of the parents. There are 3 parents here, A1, A2, and A3, and most of the children kind of look like the parents. There's a tail of dummies that are less stable than their parents. But what's lovely about this is there's a whole bunch that are more stable than their parents. Some of the children are smarter than their parents. They've figured out how to do things that their parents didn't know how to do, and in stability, that's one of those things. Now, of course, if you've got hundreds of sequences and hundreds of functions, what do? You correlate them. That's what any scientist would want to do with a data set like that. So we asked, is there a sequence-function relationship that we can discern from this non-natural family of proteins? And we were absolutely flabbergasted to find out that yes, there is a correlation, and yes, it is the stupidest correlation you can imagine, linear regression. If you do linear regression on the stability versus the sequence, where the sequence is which of the 3 parents did each of those 8 elements of the gene come from, so there's only 24 sequence elements in this set, you can say that the stability of chimera is beautifully described by the stability contributions of the pieces from each of that make up that gene. What's the consequence of that? It means with a simple model that allows you to do a simple predictive model where it's like having you and your mate have 3 children. You measure their sequences and you measure their, say, IQs. It tells you how you can design the next child to optimize a particular property. So it's like having a few children and being able to predict the properties of all the possible children. So that's exactly what we can do. We can take that set of, actually you need only about 35 sequences, and predict the stabilities of all 6,561 recombinants. And not only can you predict those, of course we know all the sequences, we just type in those sequences, send them off to DNA 2.0, get the genetic material back in the mail, and we can build them and test them. And every single one of those, it was better, was more stable. So what did we do next? We decided to apply this to a really important problem. One of the problems that we wanted to do this with was to make cellulases, enzymes that degrade cellulose. Fungi are really good at degrading cellulose, but those enzymes are not as good as they need to be in order to make biological biomass competitive with pumping fossil fuels out of the ground. We had to do better than that. So we decided to apply this idea that you could test a few recombinants and predict the properties of much better ones on cellulases. So here's an example experiment. We took 3 fungal enzymes, and the fungi, I mean, even if you have a thermophilic fungus, maybe it acts at 60 degrees centigrade. We would love it to have enzymes that are active and much more active at 70 or 80 degrees centigrade, but there aren't fungi that have provided those in the natural world. This is a typical problem for engineers. Nature does almost good enough, but not quite. And you want to get that, eke out that next 10-fold increase in activity. So we took these 3 fungal enzymes and recombined them, expressed them in yeast, and synthesized a random sampling of chimeras, used that data, the stability sequence data, to predict the most stable possible children from that recombined set. And every single one of those was more stable than any of the 3 parents from which they had been made, including the so-called thermophilic, the 2 thermophilic funguses. So we took the enzyme HJ+, which has a melting temperature of about 74 degrees, and we did our usual thing. Okay, this is now a new entity. It's a chimera of natural cellulases. And we did our usual thing where you fine-tune it by random mutagenesis. So if you go through multiple rounds now of random mutations and screening, trying to get it even more stable, we ended up with a protein, with a version of this cellulase that now loves to work at 75 degrees centigrade. At 75 degrees centigrade, the workhorse enzyme that we use now in industry from T. rice or H. jecarina it won't even function at all at 75. It's gone. But as you've accumulated these mutations, first of all through chimeragenesis in HJ+, and then through accumulating point mutations, and note this is on a half-life scale, the final enzyme is now significantly more stable. And that's very important for practical applications because you get a chemical benefit from operating at higher temperatures, which you can see here. If we look at the amount of product that this enzyme makes as a function of temperature, the wild type native enzyme from H. jecarina, that's in blue, it works optimally at 55 degrees centigrade. If you go to the thermophilic enzyme, it's slightly better, works at a slightly higher temperature. But now you look at the chimeric enzyme, it's even more active because it can go to higher temperatures. And finally, as you've accumulated the mutations, this now can work very well towards 75 degrees centigrade. This may not look like a big number to you, but when you consider that making fuels from biomass takes train load quantities of these enzymes, it's enormous, the volume of these catalysts that have to be made. Increasing the activity or reducing the amount 4-fold makes an enormous benefit. All right, so I've tried to give you kind of a flavor of where the different search strategies that nature's used that we've implemented in the laboratory, random mutations and SACs, can take you in the exploration. We can go into this space of possible sequences and use this algorithm using today's proteins as a starting point to create a whole spectrum of proteins that can be useful for humans. But this isn't where it stops, because the creativity that people have won't stop with this infinite space of possible proteins. If you think about the natural world, there's so much more out there to play with. And I think of Angie Belcher's work at MIT, for example, where the inspiration just from looking at nature, well, it's not just proteins. Proteins encode other things like minerals. Why can't we now add that to our evolutionary soup and our space of exploration? So for example, she's shown how you can evolve on the backs of viruses, the ability to template inorganic materials so that you can start to make things like batteries or artificial photosynthetic devices, all using evolution as the algorithm to circumvent the near complete ignorance we have on how this is done at the atomic level. But why stop there, right? Chemists are very creative. To genetically encode the 20 natural amino acids is fine. Okay, it's a big space to play in. But why not have more than the 20 natural amino acids? Why not incorporate 250 amino acids that all have different functionalities that the synthetic chemists have explored and utilized for so many of the products that we need today? And why not put those into the mix? And so there are a lot of creative people out there exploring the space of possibilities with a much broader range of the alphabet. So I'm going to stop here, because I know we have a nice evening ahead of us with the reception and all, and we've had a lot of good science already. Stop here and remind that sometimes the best thing to do when you want to be the designer is step back and learn the rules from the best designer there is, and that's nature, using the insight that Darwin gave us. And I'll thank a few of my students who contributed to the work that I presented today, and mostly I want to thank you for being here, and I hope you have a wonderful time.
Thank you very much, Dr. Arnold, for a wonderful ASM lecture. And we'd like to present you this one.
Thank you.
So we hope that you have enjoyed the opening session and that these informative and thought-provoking presentations have set a tone for the rest of the week here in Denver. Again, thank all of our speakers this evening, and now please join us for the opening reception. Please enjoy the meeting.